things, all of this material, I believe all of you know where. But again, that means some little bit as review what is the DNA and some of them. Talking about the living things, maybe not really all. Sometimes we usually imagine that there is theory, virus, unicellular, and multicellular organism. And when we're talking about synthetic biology, we start with the genetic element. All of the living things, I think, has a genetic element that is RNA or DNA. Let's see here, there is the, I put the simple structure virus, that is TMV, tobacco mosaic virus structure. The structure is very simple. It is a genetic element, RNA, surrounded by the protein that called capsid. And the other one is very complex structure virus. It is IV virion. Many accessories already taken by this virion. And of course, the most important is the RNA as genetic element in the virus. The real living things is actually the unicellular and multicellular organism. The plasmid that in the living thing in the world grow in the prokaryotic and eukaryotic cell. And I think we all know also what is the difference is prokaryotic cell without nucleus and eukaryotic cell has the nucleus that both containing the element itself actually constitute many in the prokaryotic. The main is chromosome, there is the plasmid, maybe some organism containing the viral genome inserted in the chromosome or transposable element that it can move from one organism to other. In the eukaryotic cell, the genetic element, of course, chromosome, some containing plasmid, any other is that mitochondrial and also chloroplast. This all is the genetic element. And again, when we're talking about synthetic biology, I think we will start to do how to understand what is the genetic element when we lyse this bacteria gently, so the DNA will mix up all of the cell, and also when we little bit observe deeply to the nucleus, there is the what we call the chromosome. Another thing here is the small circular DNA that we call plasmid. We will do our exercise maybe using this kind of plasmid, I mean the genetic element. But it is very long chain, of course, also in the eukaryotic case, very long chain of the DNA. It is chromosome plasmid, and the eukaryotic cell is the main is chromosomal genetic. It is just to imagine that the genetic element size, for example, in the genome size of some given example of organism. The first time I think that human is the biggest. What is actually the genetic element? Genetic element actually is the chain or the polymer of DNA. We make some focus here. When we put the water, there is actually the chain of nucleotide adenine, thymine, guanine, and cytosine. Just it. No other. And when we search in the database, usually we will find this kind of information. In the cells, it is the building block of the genetic material of the cell called for specific trait. So DNA is the blueprint of the life itself, and made up of EPK molecules from the nucleotide. Another component of the genetic element is RNA. RNA actually is very similar to DNA except the sugar component. DNA is deoxyribose and RNA is ribose. DNA usually has the two strands, but RNA one. DNA has thymine, RNA has uracil. This is the little bit possibly what the DNA RNA. So other is similar except thymine, the residues, and the RNA is to be uracil. The RNA. Other is what is the protein? So sometimes we hear about the DNA, RNA, and then also protein. Protein is a polypeptide, the long biomolecule or macromolecule consisting one or more long-chain amino acid residues with diverse function such as enzyme, toxin, hemoglobin, antibodies, and other. So we can imagine that protein is concentrated the polypeptide of amino acid, the chain of amino acid, which is methionine, glycine, phenylalanine, glutamic acid, and so on. And usually, this is the primer structure, the secondary structure or tertiary structure. Sometimes we found to illustrate the protein as something like this one. This is the protein. Nucleotides actually has the component. It is the phosphate group, the sugar, and the nitrogenous base. Adenine, guanine, cytosine, or thymine, also uracil, actually has the same two part of this component. And the nitrogenous base, that is 5 thymine, cytosine, uracil, only used in the RNA, and taurine, adenine, and guanine. The picture is like this one, a little bit complex for the taurine group compared to the pyrimidine. The rule that adenine must be paired with thymine, so we sometimes find the AP. Guanine must pair with cytosine GC. The amount in different DNA molecule will be about the same. AP, GC mean the pairs between adenine and thymine or guanine and cytosine. Sometimes we read the paper that this organism is high GC. It means the DNA containing a lot of guanine or cytosine residues. Sometimes the organism comprising more than 80% GC or high 80 also. This pairing between guanine and cytosine, there is the hydrogen bond, the 3 hands, and thymine and adenine hydrogen bond with the 2 hands. So it also corresponds to the when we do some optimization doing the PCR to determine the underlying temperature, something like this one. Sometimes we should consider about the hydrogen bond. DNA is double helix. The text is again that G should be paired with the C and D should be paired with the E. And you can see here the orientation. One strand is 5 to 3, another is 3 to 5. This corresponds to the carbon residue in the sugar. when we're talking about the chromosome and gene, let's imagine that it's always the DNA, and it will be very long, very long chains. A piece of the DNA we call as a gene. A gene is a sequence of DNA that encoding special traits. For example, why the flower is to be red? Because the gene correspond to the color of flowers is there is the specific sequence that will result in the protein that will give the flowers to be red and so on. So this is the gene. So the chromosome actually is the pool of many genes. In Bacillus subtilis itself, for example, there is approximately 4,000 genes. This is the example of genes. So when we get information about ATG and so on, by bioinformatics analysis, the segment of DNA can be predicted as gene or not. This is using the classical approach that gene always start by ATG, followed by one of the three kind of PAG or TG. Sometimes called as ORF, open reading frame. Sometimes the segment is not yet characterized, but the segment is have the characteristics as gene. What is it? Start by ATG and end by top order. Okay. It is the gene. Actually, still many DNA sequence that has characteristics as a gene, but not yet characterized. What is the function actually in this organism? Okay. This is the example of gene sequence. This is the already characterized. There is the ribosomal binding site. There is the start codon ATG. There is the stop codon, and we can predict what is the amino acid residue encoded by this sequence. So we can imagine actually when we want to change the amino acid residues, actually we should start change the DNA itself. I think this is the first step. What the synthetic biology part of this. We should know what is the gene and arrangement of the nucleotide is the sort of line. The code of the chromosome is in the specific order that basis order. So this is the DNA, and the structure is very specific. So actually, what is different between the bird and mice is in the DNA, in the arrangement of the nucleotide itself. So the material is the same. Our DNA is same with the DNA of rice, but the arrangement is different. That is the genetic diversity. Central dogma in the molecular biology that genetic information always goes from DNA to RNA to protein, and that now has some revision. From RNA to the DNA is possible using the reverse transcription. It's HIV virus usually from the RNA convert to the DNA and then into the chromosome of the host. Here, there is a little bit difference between the prokaryotic gene and eukaryotic gene. Prokaryotic gene, for example, in the bacteria and archaea, this is the gene A, gene B. So we can imagine that gene A is consisting of polypeptide, polymer of nucleotide. it will be transcribed to the messenger RNA. Gene A with mRNA is little bit similar. Gene B, this one. So it will be protein A and protein B, but little bit different in the eukaryotic cell. This is the gene A. The gene A consistent any exon, intron, and so on. It should be transcribed, should be processing, and then out from the nucleus on the cytoplasm, and then in this position it will be translated to be a protein. This is so very difficult actually to predict the protein residues, the amino acid residues from the eukaryotic cell compared to the prokaryotic cell, because any processing in the eukaryotic cell. Generally, genetic information flow consisting 3 steps, that is replication, transcription, translation. Replication, this is the DNA. You can imagine the double strand DNA is duplicated, producing 2 double helix. You can imagine when there is one bacteria, and this bacteria will replicate from one cell to be 2 cells. Of course, the double cells should have new chromosomes, and this process to make duplication of DNA is called the replication. So DNA cell can be replicated by the apparatus that exists in the cell system itself. Other is transcription, transfer of the information from DNA to RNA. DNA to RNA, the product can be messenger RNA. This one will be translated protein. The RNA, it will be transferred RNA to help ribosome to do the translation process, and mRNA it will be used to construct the ribosome. The program of transcription. And this is the example. Translation, synthesis of specific polypeptide from the messenger RNA. It will be made using the 3 bases of RNA that's called codon and genetic codon alpha is the protein synthesis. And system of protein synthesis include the ribosome, protein, ribosomal RNA, tRNA, and enzyme. So actually the genetic information flow is very complex system. Let's see one by one. DNA replication, use the semi-conservative model. This is the whole DNA. They replicate to be 1 double strand DNA to be 2 double strand DNA. It is also, when I learn about this one, I little bit very difficult to imagine about this one. I always imagine that when there is a chicken and the chicken has an egg, and egg to be small chicken, but the DNA is not like this one. The old one, the new one still containing one pair from the old one. This is what we call semi-conservative. Semi-conservative. The new DNA actually containing the old strand and the new strand. Okay, this is the DNA replication. I think our cell will be very busy to always do the replication when you want to replace the old cell, the guide cell, and so on. It is the process of replication. Another thing, that is transfer of information from DNA to RNA. Again, the product is messenger RNA, transfer RNA, or ribosomal RNA. DNA must be copied to messenger RNA, and mRNA goes from nucleus to the ribosome in the cytoplasm. When we imagine about the eukaryotic cell, The example is our cell. In the nucleus, there is the DNA, and the DNA, the information want to be expressed to be a protein. The process should be through the step that is synthesis of mRNA. mRNA, the nucleus will go out to the cytoplasm, and in the cytoplasm it will be read by the ribosome to be translation. So process the reading of DNA to the mRNA called to transcription. Okay. Next step is translation. That is the synthesis of specific polypeptide from mRNA, not the RNA, not rRNA, because different function. The product of transcription is 3 kinds of RNA. mRNA is translated in order 3 nucleotides, and translation of mRNA begin at the start codon AUG and stop codon it correspond to the characteristic of the gene. ATG will produce the segment of mRNA as AUG, and the stop codon will produce as the segment in the mRNA, and it will be recognized as stop codon when the process translation is happened. This is the codon. Actually, we have 20 amino acid, and the specific amino acid, there is the specific codon. Even not one amino acid, one codon. Sometimes one amino acid is put more than 2. For example, the leucine, there is the 4 codon. So it can be implicated that even though the DNA sometimes is different in the protein residues, it is not always different. Okay. This little bit animation how the translation is conducted in the cytoplasm of the cell. This is ribosome. Ribosome will bind to the mRNA, the start codon AUG. Ribosome will call to tRNA to bring the methionine, and then ribosome move forward. This is the oligonucleotide methionine, and with the next codon, they will call to the transferring RNA to bring specific amino acid K. So this process will lead through to the mRNA. So we can imagine that the polymer or polypeptide is synthesized K. Polypeptide synthesized. Ribosome meet the stop codon. It means no translation anymore, translation process anymore. Then there will be amino acid chain here. We call as protein. That is the process of how the genetic information from the DNA to be protein. Again, the transcription and translations. Okay. As the resume of the genetic information flow, we can imagine here that there is a gene like this one. So the mRNA should be like the sequence, and the protein will be here. This is the methionine. 3 codon here is leucine, and so on. So again, we can imagine when we want to change the amino acid residue here, we can start from this one, regulation of gene expression. Actually, the gene in the chromosome is not any time expressed by this organism, but the expression of specific gene has the regulation. We can see that the gene expression is controlled at the level of transcription. We know that some gene has strong promoter, weak promoter, and so on. It will control the level of transcription. The strong promoter means that the gene is very easy to be recognized by the RNA polymerase. So there is a level of control in the transcription, and there is the processing also in the eukaryotic RNA splicing to remove the intron, modification, transporting, and translation. It's any specific rate, different gene, different regulation. In the prokaryotic cell, of course, the transcription coupled with the translation, because it's happened in the cytoplasm. And the translation, the control is similar to the transcription. Ribosome will translate it not in the same rate, one gene to other gene. Gene product itself consisting the structural gene. The function is substructural protein, enzyme, and regulatory protein. Sorry, actually not this one, because structural gene. Regulatory protein is in this position. The structural gene usually express constitutively without any types regular. In the now experience, in the past also not always constitutively. But the other is regulatory gene, that the function is a specific protein and regulation of other gene expression by binding to particular site on DNA. So there will be an example how the gene regulation. The classical example of gene regulation is the lab operon. We will use also in our lab exercise often. Yeah, a lab operon. We know in E. coli chromosome there is a piece of DNA that already characterized there, encoding the lab I gene, lab Z gene, lab Y, and lab A. This gene is correspond to the utilization of lactose, a kind of carbon sources, How this gene is expressed, there is a specific order. Not any time this gene is expressed by the E. coli, but any specific situation, any specific regulation. This gene consisting this operon, there is another term, operon in one or more gene that's controlled by one promoter. It is the called the operon. This is only one, a single mRNA, but consisting of more than 2 genes. This operon. There is in front of the promoter region, there is another gene that is lab I, and this lab I is as a regulator. This lab I will regulate the expression of 3 other genes here. This is the regulatory gene on the control region, and this is the coding region. Actually, utilization of lactose involves these 3 kind of gene. How we can observe this phenomena? When we grow up E. coli on, this is the function of the 3 types. When we analyze the promoter or operator region, we can see in the downstream of the lab overall, there is the area in this one, in the purple area is area where the RNA polymerase start to transcript. This area overlap with the operator region. Operator region in this one where the Rna protein is occupied. So we can imagine when there is a Rna protein exists in this position, RNA polymerase could not start the transcription. This is some little bit explanation. There is no lactose. Where there is no lactose, what happens? Of course, lactase is expressed constitutively. The lactase will make tetramer, and the tetramer of this protein will occupy the operator region. Lactase come into this region, RNA polymerase no place anymore to start transcription. So this gene is not expressed. Cell not containing lactose, lac overall not expressed. It is very logic because no lactose. Why we should produce the lactosidic and other? But when the cell exposed by the lactose, what happens? The lactase will bind to the lactose. So lactase will not come to the operator. Then what happens? RNA polymerase can start transcription. So when the lactose exists in the cell, the lac operon will be expressed. So lac operon will be expressed only when lactose is available. It is a type of regulation. So the cell will be very efficient to face in the environment. This is the lac operon. I think the other explanation is like this one. Regulatory gene, lac pressure will express and come to the operator sequence. What happened? This is the RNA polymerase. They will stuck in this position, could not proceed to the transcription of the lac operon. So no transcription anymore when the cell is not containing the lactose as the inducer. But when the cell has the lactose, lactase again they will bind the lactose, and RNA polymerase can proceed the transcription. Kind of protein will be expressed and correspond to the metabolism of this lactose. So that is the little bit simple regulation in the genes expression. So sometimes we, in my experience, when we assay in the laboratory, we put the chitin to induce the chitinase. We hope that chitinase actually will express in the nature, but when we release in the nature, the environment, no chitinase anymore. So what happens? There's no chitinase production. So when we want to use this microbe for biological control, for example, it's no effective anymore because no inducer for expression of the specific trait of this microbe. Okay, I think complexity in the gene expression. This is the one part of when I was my doctoral in Spain. When we challenge the Bacillus subtilis with the antibiotic, actually involve hundreds of genes to overcome the antibiotic challenges. For example, vancomycin, there will be any cascade. This is mean 1, 2, 3, 4, 5 gene cascade, and then go to the sigma factors, and the sigma factor will induce the expression around 150 gene. So actually again that the gene expression, the regulation and so on is not as simple as we imagine, but it's also a very complex system. If I look at the lab apparat itself, it will be correspond to the availability of the glucose, CIMP, and other chemical components in the cell. I think that is my presentation. Before I close, maybe any question is okay.